Good afternoon, everyone. How are you doing? It's Friday, and I'm sure you'd much rather be any place but here right now. Uh, Thank you for making the effort to actually get here. Let's do that. Don't do that. You need that one. Okay, still waiting for a couple people to show up, but so our overall goal for this afternoon is to finish chapter 27, and then maybe spend some time thinking about 25, 26, and 27. What are the things I really think you should be paying attention to as you get ready for this fourth exam? And while we're waiting, I'm sure people have noticed that online quiz is now available as well, and you'll have till Sunday, I think, at 3 o'clock in the afternoon to get that done. Recommend maybe taking a stab at it on Saturday, figure out what you did right, figure out what you did wrong, and then come back and get a much better grade the following attempt, if that's the way you want to approach it. But the idea, of course, is when you make it a little bit lower stakes, there's a little bit less stress, and therefore you're more likely to remember the material as you're going back and reviewing it. Do you guys have any questions of me while we're just sitting around and waiting? No? Okay. Well, let's just get going then. Because I am recording it. We'll just put this up on Blackboard, and you'll notice, thank you. Somebody reached out to me and asked me to make sure that all of the recorded classes are up on Blackboard, and I just checked that. That's why I'm starting late. I checked to make sure everything was there. So if you want to go back and look at those, they're there for you.
I do have a question, sorry.
Yeah, fire away.
I noticed that on the announcements you put up our two groups for lab.
Yeah.
So starting Monday, we're going to be meeting in classroom. Well, A, what classroom? And B, are we starting at 5 or 5:30?
Okay. I'm going to actually send up final notifications on Friday on Monday, because we haven't actually been told. I can tell you our classroom has changed. I don't know what it's changed to yet, number one. Number two, I can tell you that there are still people who are trying to change their schedules. So some of the groups may change. I have a size limit I can put inside each class. So right now, I just sort of went down the list and said, we're going to split you guys into 1 and 2. But I've had some requests and I'm trying to switch some things around. But yes, you're going to be up on the 2nd floor of the Health and Life Science building on the West Campus. Probably start at 5:30, maybe a little bit later. It probably won't be a short evening for any of the 3 major content evenings. But the 4th evening is more along the lines of review of everything we've covered, so you can be ready for a practical.
Okay, perfect.
Cool. So yeah, I'm still working in the dark just as much as you are, but I hope to have it all squared away by Sunday evening.
Sounds good. Thank you.
Of course. Thank you for asking, Gia. Okay, so let's figure out what acid and base balance mean. And since the beginning of the semester, I've been sort of drawing this, what looks like a childish cartoon for you. And it looks sort of kind of like this, right? Right? It goes up and down like that. And we call this homeostasis. And remember that now, because we're inside chapter 27, what this means is this is the pH of the interstitial fluid of your body. And probably more specifically, the pH of the interstitial fluid of your body as being measured inside the hypothalamus. Now, that's both partially correct and partially incorrect. Your body can withstand these extremes because of buffer systems that are correct. So whatever is being measured truly isn't necessarily what's being experienced, say, in like your big toe. How do we actually maintain this range? Because that's the hard work, right? You stay outside of this range for any period of time, it will lead to death, regardless of you going below 7.35, going above 7.45. We can discuss this evening, introduce at least the names of buffer systems. There's only one real buffer system you guys have to have tattooed someplace on your body at this point in time, and that's the bicarbonate buffering system, okay? So when I say bicarbonate, you should immediately think carbonic anhydrase, yada, 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 blah, blah, blah. But it does give us an opportunity to review what the role of exhalation of carbon dioxide actually means. How do we transfer most of that carbon dioxide to your lungs? We transfer it as bicarbonate inside of red blood cells or around red blood cells, right? And you exhale that out, and that allows us to get rid of materials that can make hydrogen protons. And lastly, here inside that, we just finished chapter 26, but the important thing to remember there is that the different locations, and primarily we're thinking about the proximal convoluted tubule and primarily the early distal convoluted tubules, where the role of maintaining hydrogen proton excretion, what ultimately is sent out of the body in urine, is what you're actually paying attention to. So these three major mechanisms are what are helping you maintain a normal acid-base balance. There's a buffer system you probably really haven't heard about before, maybe. I don't know. Maybe you have. There's something funky going on this evening with the presentation, so this is probably going to crash on us again. But what I want you to notice is that proteins can actually act as a buffer. And it's not often people discuss this with you. We tend to think of it more along the lines of its role in osmolarity. But if you think about what a protein can do, it can actually accept and release charge depending upon what's needed. So in some instances, a protein buffering system, okay, the carboxyl group of a protein buffering system can actually act like an acid, which is kind of interesting. It gives you a different way to think about, say, albumin, fibrinogen, all of those structures again. Let's see. Obviously, when we think about hemoglobin inside of a red blood cell, this is probably the best example I can give you. Because remember that switch, depending upon where we are we actually inside the lungs or are we inside, say, tissue cells? What's happening as this occurs is the shape of hemoglobin is actually being changed. And notice here, just in this lower piece, we've been through this a million times, okay? What's changing here is we're describing what's actually happening to hemoglobin. And by adding that hydrogen proton, okay, we now have reduced hemoglobin. And it's not often you actually hear proteins being described as either reduced or oxidized. So here's that carbonic acid bicarbonate buffering system again. So we just experienced it. So this means Mother Nature has multiple layers of systems that can actually buffer simultaneously. So if you've ever wondered why the range is as tight as it is, 7.35 to 7.45, it's because all of these systems are functioning simultaneously. It isn't like you choose, well, we'll use this one this time, and we'll use that one next time. No, they're all functioning at exactly the same time. And I don't think I need to actually go through this with people in the class. However, here's a new one. And this is primarily an intracellular phosphate buffering system, okay? Where we have something that's called dihydrogen phosphate, H2PO4, and monohydrogen phosphate, HPO4 minus. And these are used such that any hydroxide ion, which is a strong base, will now bind to a weak acid, that dihydrogen phosphate. And what's going to happen is it becomes now monohydrogen phosphate, which is now a weak base. So we have taken a strong base, we've converted it into a weak base, and that means the other buffering systems can now act to maintain that tight pH range. And look at what you get out of it. You get water, okay? So inside the cell itself, this is a mechanism that can be used to maintain the total water component of intracellular fluids inside of the cell itself. And that's one of the things I want you to pay attention to, is notice I'm talking about the phosphate buffering system primarily being an intracellular system. It does act inside your urinary system on filtrate, but primarily when we're thinking about it, it's an intracellular buffer. Okay, carbon dioxide. Think about that last exhalation when you read a question on the exam and you had no idea what it was asking. Remember, pause, take a deep breath, inhale, exhale, and what you're actually doing, of course, is you're getting rid of carbon dioxide, and your brain is going, good, now you're feeling better. And while this is actually happening, you're making first carbonic acid, and then you actually have more bicarbonate. Okay, and what I mean by that is you have more ability to then convert the bicarbonate ion into water and carbon dioxide, and then you can exhale more carbon dioxide. And of course, this is what's actually happening inside your entire body, and we're going to use, say, bicarbonate here as a response, but you can see a stimulus is going to cause a blood pH change. We have an increase in hydrogen proton concentration. So what that means, of course, is if you have more of these, the pH value drops. Don't be one of the students who falls for that type of question, okay? And the central chemoreceptors inside the medulla, as well as the peripheral chemoreceptors inside the aortic and carotid bodies, will now change the respiratory group. And notice it's the dorsal respiratory group, and that's going to cause more forceful exhalation of carbon dioxide. So it's no different than what we learned back inside chapter 23, I think. And just pause for a second. How long ago does chapter 23 feel? Just think about that for a second. Exactly, your brain has sort of created this time warp, right? Okay. So the mechanism is the same as the point, but in this chapter, we're thinking about, exactly, go ahead, exhale. Okay, well, you're thinking about what that actually means. And this is a feedback system. So what we're looking at, of course, is the production of carbon dioxide to become bicarbonate creates the feedback loop. To bring us back into homeostasis, the blood pH or hydrogen proton concentration needs to get back inside that the expected range. Probably the trickier one, or trickiest one, is probably the kidney's role at moving hydrogen protons, right? And excreting hydrogen protons in urine removes a non-volatile acid. Carbon dioxide, bicarbonate, these are gases, they're volatile. Hydrogen protons, they're not volatile. And remember, I said a moment ago, the proximal convoluted tubules and the collecting duct, these are going to be the locations where we actually secrete hydrogen protons into that tubular filtrate. So the takeaway point here would be, if we're looking to monitor or figure out how, imagine if you will, we will have to do this in a few minutes, your lungs aren't functioning better. It would mean then that your kidneys at the proximal convoluted tubule and the collecting ducts would have to secrete more hydrogen protons now into the filtrate. Your kidneys fail, that means your lungs are going to have to move more carbon dioxide out of your body. Okay, so this interplay, this acid-base balance, means that you have two major mechanisms that can control pH inside of your body. And this is that example that we saw inside chapter 26. What's going on here, of course, is here is that collecting, so here is an intercalated cell inside the collecting duct. And what's happening is the process of making bicarbonate, okay? So here's carbonic acid, here's our bicarbonate here, here's our hydrogen proton, and using a proton pump, we push that hydrogen proton now into that fluid. But notice what I'm doing. I'm telling you the cell name that's actually doing the work. So for regulating a metabolic response to pH changes, we would use intercalated cells in the kidneys, in the collecting duct, to secrete hydrogen protons. Woohoo. Now, remember I said a moment ago you might use a phosphate buffering system here. Well, that's because in this case, we have ammonia, and ammonia needs to be converted into urea. And we want to make sure that this actually happens. We won't worry about it in terms of a testing type question, but the idea, of course, is hydrogen protons can then actually work in additional systems. Okay, and now you actually have those hydrogen protons here. When and if, see, we won't do this lab in this lab. We won't do this part of the lab when we actually go into the buildings next week and the week after that. But when you were supposed to urinate on that dipstick, literally, you actually have a range, and the range goes down as low as 4, and in some of them it goes as high as 10. What you're supposed to see is that depending upon the chemistry of what's happening inside of your body, you may actually have a very high concentration of hydrogen protons, and that would give you a low value in your pH range. So this is the sort of the summary of everything I just sort of walked us through. And I guess if you're sort of loath to read chapter 27 because it just looks like a bunch of, gobbledygook with ranges and all of that, I would probably start here thinking about what Mother Nature is doing. Mother Nature's using carbonic acid bicarbonate buffering systems for extracellular fluid. She's using phosphate systems for intracellular fluid and urine. She's using exhalation of carbon dioxide to get rid of, exhalation of carbon dioxide to do two things. With increased exhalation of carbon dioxide, the pH will rise because there's fewer hydrogen protons. With decreased exhalation, the pH will fall because you're not getting rid of those hydrogen protons. So look at this. This means we can think about, we have a switch. We have a mechanism we can work with. Likewise, inside of the kidneys, the renal tubules will secrete hydrogen into urine and reabsorb bicarbonate so it actually isn't lost. It isn't really a concern for us in terms of reabsorbing that bicarbonate. We have plenty of it inside our bodies, but what is most important is you remember that the kidneys are where we will use those intercalated cells to get rid of hydrogen protons. Okay, so you are this bag of salt water that has a specific range, and we can describe you depending upon what you see when you walk inside a patient's room. You look at their pH, you're like, it's below 7.35. Well, it's below 7.35. It means they're becoming acidotic. But you don't know if it's respiratory acidosis or metabolic acidosis. You don't know if their lungs are failing them. You don't know if their kidneys are failing them. Okay, so this means you have to set about asking a series of questions. Likewise, you take a look at their values and you say, oh, pH is above 7.45. Okay, that's another problem. Okay, but again, you don't know if it's respiratory alkalosis or you don't know if it's metabolic alkalosis. And notice what we are measuring here. What we're measuring is a blood pH, which is we know what the range is, due to excessive retention of carbon dioxide, leading to excesses in carbonic acid. Okay, so we're not getting rid of carbon dioxide is the point. When we think about what's happening to respiratory alkalosis, the blood pH is going to rise due to excessive loss of carbon dioxide. So we've gotten rid of too many, which means you have more of an alkaline environment. Neither of these are good if they're sustained for any period of time. But notice what we've done here. We've looked at the pH value relative to the amount of carbon dioxide. When we're trying to assess what's happening at the metabolic level, we're looking at the levels of bicarb. Okay? You're like, well, what's the difference? Well, the difference is, are the kidneys actually collecting, producing, retaining that bicarb? And in metabolic acidosis, the arterial blood levels bicarb fall. In metabolic alkalosis, the arterial blood levels of bicarb will rise. Okay, we measure these. We measure these in terms of specific values, right? What is the partial pressure of the gas of carbon dioxide? What is the pH of the fluid? What is the milliequivalent concentration of bicarb? And these are the three sets of values you want to start thinking about. You're like, but I'm just thinking about it now. When will I have time? Oh, I don't know, tomorrow. Okay, maybe. What's the complete definition of respiratory acidosis, right? So the take-home quiz, I think I'm dying of something on your take-home quiz, and you guys are supposed to save my sorry ass. My advice, of course, would be just let me die. Okay, but you're not going to do that, right? Respiratory acidosis, you have increased partial pressure of carbon dioxide above 45 millimeters of mercury. You've decreased pH below 7.35 if there is no compensation. And here, when we start talking about what compensatory mechanisms are, is if your lungs are failing you, your kidneys compensate. You excrete more hydrogen protons, and therefore you increase reabsorption of bicarbonate. Okay, and that will work as long as your kidneys are up to the task. Exactly. Okay, to keep the partial pressure of carbon dioxide within its normal range. So once you learn one definition, right, respiratory acidosis, means you can look at respiratory alkalosis. You have a decreased partial pressure of carbon dioxide. It's below 35 millimeters, and the pH is above 47.45. Renal decreased excretion of hydrogen protons is what the kidneys would have to do. So let's go back. Think about how I tried to show it to you, right? you have a range. So let's go 7.35 to 7.45. 7.35 to 7.45, right? So 7.45, 7.35. Okay. And anything that's above 45 millimeters of pressure is going to suggest to you that this is now a respiratory acidotic state. So you draw this out, you have your ranges for, go like this, put a line here, put CO2 here, CO2, and I'm gonna put PCO2. That's supposed to be a C, it looks like an L, sorry about that. PCO2, so it's 45 to 35. And you're like, wow, that's so cool. I just recognized something. The numbers are close to pH values. Okay, did anybody else see that? No? 7.35, 7.45 is the range for pH of your interstitial fluids. The partial pressure of carbon dioxide is 45 to 35. That's the normal range. And anything outside of that now, once you understand this, this helps you decide what are you looking at. Okay, so increased partial pressure of carbon dioxide above 45. So put arrow up, put pH value down. Therefore, we know we have respiratory acidosis. Also, if the question asks for, is there a compensatory mechanism? And the answer would be yes, if this patient stayed in this, distressed state for a period of time. The kidneys would increase the excretion of hydrogen protons and then try to collect more bicarbonate. And that's the way you want to think about acidosis and alkalosis. Does anybody know anybody who's working in a hospital now? No? Yeah, okay. Ask them, so are you working in a hospital now treating patients? Yeah. No, okay. So ask a seasoned
Not
veteran.
anymore.
Yeah, okay. So ask a seasoned veteran to show you the back of their ID, right? And what you're going to see on the back of their ID is exactly this thought process. Because what usually happens when people are stressed is they forget 7.45, 7.35, 45, 30, 35, and then the last one's going to be the bicarbonate, which is 22 to 26. 22 to 26 milliequivalents. And when you have these three pieces of information, you can then figure out what's happening to your patient without fail. So what I want you to do, of course, tonight, when you don't want to do it, is just think to yourself, hey, if I had a patient in front of me that had a pH value of 7.28 and they had partial pressure of 48 millimeters, do I understand immediately that they're in respiratory acidosis? And if I did have to think about their compensatory mechanism, they would have to increase the amount of bicarbonate being reabsorbed and excrete more hydrogen protons. And that's the way you want to approach solving these types of problems. And believe me, all of these problems are in your future, not only because this is what actually kills your patients, but also because these are the types of test questions people love to give students. So we already did respiratory alkalosis. Let's take a look at metabolic acidosis. Notice I said a moment ago, we have a specific range for bicarb. It's below 22 milliequivalents per liter, and the pH is decreased below 7.35. So in this case, metabolic means I have two values that are going down, and that's actually how you figure out what's going on. Okay? Literally, when I'm looking at a write-up of somebody, I'm like, okay, are the two arrows going down? And if the two arrows are going down, I have a pretty good idea what's going on here immediately. What's interesting, though, is the respiratory response is to hypoventilate the individual, okay? Which is now going to do what? Okay, if you read closely here, it's going to say, whoops, get this out of the way. Sorry about that. No, you get out of my way. You won't move. Get down there. Forget about it. Okay, I don't know what you guys see, but you guys are flickering all over the place here. Okay, but the pH should be returned back to the normal range, but bicarb is going to do what? What does it say there?
Be high.
I'm sorry, Brooke?
It says it will be high.
Yes. Would you have expected that?
No.
Well, in some ways you would have, right? Because bicarb can bind hydrogen protons. So it needs to remain high in case more hydrogen protons are being made. That's the only thing that I kind of want to stress here, because remember when we did the bicarbonate buffering system, remember it goes H2O plus CO2 gives us carbonic acid. The carbonic acid then gives us hydrogen protons and bicarb. And if you have more bicarb around, it means it can bind any spurious hydrogen protons, and that's what you actually have when you're in acidosis. Okay? Cool. Now the last one is this, okay? Here we want to pay attention to the fact that pH has gone above 7.45 and 26 milliequivalents is a high value. So in this case, 2 arrows are going up. And that's how I know the patient's at metabolic alkalosis. If you can remember that, you're going to find out, or this is what actually happens here is, what's happened is individual, probably through either drug overdose or something like that, vomiting, gastric suctioning, or specific diuretics, these will cause a major shift in the bicarbonate concentration, as well as a rapid rise in pH there. And what's going to happen here, of course, is again, you're going to have to figure out hyperventilation or hypoventilation. I think there's something wrong with this figure here. Okay, I need to go look at that, get back to you guys. Whoops, that's the end. We'll take a look at something before we actually move on. So for metabolic acidosis, let's go back here, folks. There's a mistake. I knew there was something wrong there. For metabolic acidosis, that should actually say low, okay? Right there, that should actually say low. And metabolic alkalosis should be high. There we go. Good, okay, cool. Glad I caught that, because that would cause creation of problems for you. Okay, and the idea, of course, is the pH range is what the tell is. Okay. Woohoo. But that's chapter 27. And that's, so really all we did is give you something to think about in terms of if we give you a description for pH, bicarb, and carbon dioxide partial pressures, you can do a series of if-thens, and those if-thens will help you find out if you're going to save my life or kill me. Okay? Not that you want to do either, but you know, whatever. Okay, so let's go here. Okie dokie then. Does everybody see the whiteboard in front of them? Yes, no, maybe? Cool. And we have (25), 26, 27. Right, we just finished 27, but we'll make sure we spend some time thinking about that. So 25 is metabolism, and remember metabolism, it seems like a lifetime ago you were thinking about it, but remember this is 1 6-carbon structure being brought into a cell and being converted into 2 3-carbon structures, which are then, if oxygen, let's go like this, if oxygen is available, we make pyruvic acid, so I'll put the P for pyruvic acid. If I go over here, I'm going to put an L here, I'm going to put an O2, and I'm going to put no O2, okay? So this is fermentation over here, and we make a certain number of ATP molecules, but we have a reduced molecule now that will be stored inside your liver or travel to your heart where it will be metabolized there. Pyruvic acid, of course, will be modified when we go inside the mitochondria to give us acetyl-CoA. So P becomes acetyl-CoA. You want to pay attention to where that's happening and what's actually going on there. Okay, what is the decarboxylation? Why is that important to understand? And then once this has occurred, now we're actually in the Krebs cycle or citric acid cycle, CAC. We're not asking you to memorize all of the steps of, say, glycolysis. We're not asking you to memorize all the steps of the Krebs cycle. We're not even asking you to memorize all the steps of the electron transport chain. We want you to remember the citric acid cycle makes a certain number of, say, NADH plus H plus molecules. And I stress the H plus molecules because this is a specific type of transfer. It's a proton motive force that will create the energy required for chemiosmosis when we make ATP at the ATP synthase. Okay, so we need to know how many NADH plus H pluses, how many FADH2s, okay, and the significance here is, even though I'm not mentioning carbon dioxide, we just spoke about carbon dioxide, right? So the more metabolically active a cell is, the more carbon dioxide that cell makes. And this means then that that's why they tie all of these chapters together. Okay, that's why we put 25, 26, and 27 together. So as you change metabolism, you're going to change what you would actually see inside your patient, right? Ultimately, though, when we finish off chapter 25, you got in here, you've made, what is it, 30 to 36 molecules of ATP, right? Then we talk about, well, what are some of the other mechanisms that can be used? And you can use intermediates of either glycolysis or intermediates of the Krebs cycle that send material to the electron transport chain. If I'm you, though, and you understand, I'm assuming you're going to spend most of your time saying to yourself, what the hell is going on here? I mean, you're just going to try to memorize the values for what are the products. And that's a pretty good place to start. You're not a biochemistry student. We don't expect you to be able to draw these mechanisms out or anything like that. But you should know, how much ATP do you make in glycolysis? How many molecules of carbon dioxide, right? But when you're thinking about how it's regulated, how metabolism is regulated, there are two tables, one for pre-absorptive and the other for post-absorptive mechanisms that change the utility of intermediates to make ATP. What does insulin do? What does thyroid hormone do? What does epinephrine and glucagon do? This is the end part of chapter 25, but there were these two tables inside the pre-absorptive and post-absorptive pieces that discussed specifically what those hormones do. Nudge, nudge, hint, hint, wink, wink. Okay, chapter 25, done. I'm recording this, right? Somebody would have still told me if I wasn't. Okay, right, chapter 26, you're like, oh my God, that feels like yesterday. And we were just talking about it yesterday, right? Where the major mechanism for kidney function is blood flow to a nephron. Yay, and you're like, he's gonna draw that stupid cartoon again. Yes, in fact, you wanna practice drawing it, not because you want to, but because you know fine well there's gonna be specific locations on it that you need to know the function of. Change color here, where'd you go? A, E. Notice what I'm doing this evening. Okay, and then ultimately that flows out. Where what you want to make sure you're thinking about is, if we thought about glomerular filtration rates here, so that we end up making filtrate that passes through the proximal convoluted tubule, down the descending limb of the loop of Henle, up the ascending limb that is now water impervious in the juxtamedullary nephrons. As that ascends, there's supposed to be another layer here. Didn't draw it in, sorry. Put it in a second, right? As that's moving, notice there were specific regions where there was regulation by ADH, A2, ANP, as the three major hormones that regulate the rate at which materials are either reabsorbed or secreted out of the kidney. Normal cortical nephrons, there's very little regulation. Most of the reabsorption occurs inside the proximal convoluted tubules. Remember, at any given moment when you become more and more dehydrated, the concentration of ADH gets higher and higher, and you shift then the responsibilities to the juxtamedullary nephrons, where the cells now begin to increase the number of aquaporin 2 channels to reabsorb more and more water as you increase the amount of sodium, potassium, ATPase, collection of sodium, potassium, and chloride. So that's the basic mechanism. That's the stuff you really want to make sure you understand before you sit down to take the test. And I think the practice exam, it's pretty much 26 and 27. There's very little 25 on it, if anything at all. That was more of your, make sure you put the answer to the question. If you've got 100 molecules of glucose, can you tell me the numbers, right? Why would I ask you a question like that? Well, it's to make sure that, number one, you could figure out how many ATPs and all of that. But notice here at the end, I said, if you needed to modify that, you would use which hormones in the pre-absorptive state, which hormones would you use in a post-absorptive state. Cool. Now, chapter 27, you're like, okay, we just studied that. In which case, this makes you a bag of salt water, right? And what's really important about chapter 27 is all of the mechanics we've studied all semester long. Okay, how do you maintain a specific volume of water that is extracellular or interstitial? I stands for interstitial here. Okay, or intracellular, so I'll put intra. And you're called salt water because there's a specific concentration of different ions that are outside here versus inside here, where the heres are either extracellular or intracellular. And that's honestly the way you want to, if I'm you, if I'm thinking about what it means to be alive and what Mother Nature is actually spending her energy doing, she's spending her energy separating these ions from one another so that, one, we can make a resting membrane potential across all those cells, but two, to make sure that when they change their location, they do the job they were supposed to do. And this is why in AMP1, I stress that when people tell you that homeostasis is equilibrium, it's not an equilibrium. It's an engineered disequilibrium of the concentration of ions between membranes inside of your cells. And that's really where the hard work of understanding what this does, because anybody who treats somebody for, say, concentrations of hydrogen protons, okay, if you give them a non-specific hydrogen proton pump inhibitor, it means now you've stopped all of the hydrogen proton pumps in different parts of the body. You may want to stop that inside their stomach, but you never want to stop that inside their kidneys, right? If you stop the movement of hydrogen protons inside their kidneys, you immediately put them into a respiratory compensatory mechanism, right? So when you practice acidosis and alkalosis situations, really what you want to be thinking about is what would happen if these failed, if the person was in a fire and they can no longer respire effectively. It means now that their kidneys have to pick up all of that additional responsibility, okay? And that's what actually drives, in many cases, the failure of, say, questionable kidneys, right? You add a specific stressor to them that wasn't present before, and now it pushes the mechanism too far. So I'm gonna just open it up for sort of general questions. I think, you know, hopefully you can see. Yeah, I know. I didn't walk through what it means to be dehydrated here, but remember, each one of those flow charts I showed you last night, well, all they did was increase or decrease the volume of either a water or a salt inside of you, which makes you a bag of salt water. What we did this evening, though, is we paid attention to what is a pH range, right? And then notice we paid attention to the pH of normal ranges. Let's go 7.35 to 7.45. And the two new tools we paid attention to this evening were, what is the partial pressure of carbon dioxide, and what is the milliequivalent concentration of bicarbonate, HCO3 minus. And remember, this is 35 to 45. And partial pressure, milliequivalents of bicarbonate are 22 to 26. Right? And this means that once you know those numbers inside your head, any deviation outside of those ranges is telling you something. Okay? So what questions do you guys have of me? None?
I have a question, Professor.
Sure, fire away.
For example, if there's a patient which has kidney failure and his kidney cannot process the sodium.
Yep.
What happened?
Well, so it isn't so much the kidneys aren't processing the sodium, but you could, so let's imagine there's a mutation inside there, any one of the sodium transporters inside the kidney. And let's start there, okay? If we can't sequester sodium inside the body, it means the individual runs a high likelihood of dehydration, because if you're not storing the solutes inside the body will no longer, osmosis will not occur to dilute that solute, okay? And that means water will then leave the body. So patient would probably put on a saline drip to make sure they stay inside the same range. That person, if they truly did have a mutation inside those transporters, they probably wouldn't make it too far in life, because if they have a mutation inside those transporters there, they have it in other places. Does that make sense?
Yeah, thank you.
Of course, no problem. Okay, so go back, finish your reading. Unfortunately, I can't say don't read something. You know, you do want to, even though I tried to focus you on carbohydrate metabolism here, don't forget about what causes, say, lipolysis versus lipogenesis, what causes gluconeogenesis versus, I don't know, glycogenolysis. You know, you want to make sure that there's a reason why I said you need to know what insulin is doing versus glucagon. Okay, nudge, nudge, hint, hint, wink, wink, right? From my perspective, probably the most important information is inside chapter 26. Make sure you know glomerular filtration rates. Make sure you have a general idea of what the proximal convoluted tubule is doing versus, say, the early distal convoluted tubule or the ascending limb of the loop of Henle. Okay, and if there's a question on your exam that's speaking about the vasa recta, you know immediately you're thinking about the juxtamedullary nephron as opposed to the cortical nephron. You want to read closely enough that those cues are telling you where you are inside the kidney. Remember, all materials that are going to be secreted out, they travel through the column to the minor calyx, to the major calyx, finally to the ureter after it's traveled through the ileum, now to go into the bladder. And remember, there's two layers of reflexes there. There's an involuntary sphincter and there's a voluntary sphincter, right? The involuntary sphincter you can't control. The voluntary sphincter you learn to control. And that's what allows urine to actually exit the body through the urethra. Don't confuse ureter and urethra. Don't be that student who isn't reading closely enough to, you know, make sure that they know, you know, where things are traveling. And as usual, I mean, I'm grading your, what am I grading? I'm grading my Bio 121 lab books. So I'm not doing anything of yours workbook yet till you give me your next set of take-home quizzes. But I'm around is the point. I'm not going away or anything like that. So if you have questions, email me. Okay? Okie dokie, folks. Go have a nice stiff glass of water and grab your book. And I did the experiment for Elizabeth last night. For dinner, I didn't feel like cooking anything at all, so I just ate a bag of potato chips. So
Crusty eyes?
Oh yeah, crusty eyes. And I figured I would report out. You know, yes, I did the experiment. Okay. Now, of course, I could have been biased because I thought I already knew what the output was going to be, but I was like, huh, I wonder if they'll even, yeah, okay. But yeah, so crusty eyes.
All right.
Okay, so have a wonderful night, and I will see you guys on Monday. Hopefully you'll have a good test experience. You'll hate me only a little bit more than you already do. And I will see some of you on Monday evening, and I will see some of you on Tuesday evening. As soon as I get all of that sussed out, I will put that up on Blackboard and I'll let people know it's there. Okay? Thank you for your attention. Somebody loves you somewhere, just not here right now. Bye. Woohoo! Stop share and